Welcome back to another 461 essay video. Today we're going to be talking about the nephron and a little bit more about the kidney. We just have a couple topics to cover and it shouldn't take very long. But we're going to start a little bit about reabsorption and how that works and how solutes and water are reabsorbed in the kidney tubules. So we're just going to talk about how sodium, glucose, water, chloride, calcium, and potassium are reabsorbed. Basically, how that happens is I have a nephron already kind of drawn out. I kind of drew everything out just beforehand, just so we can walk through it together. But where reabsorption occurs, so if we look at the definition of reabsorption, reabsorption is when we're going from, we have solutes that have already been filtered, or solutes in water that have already been filtered into the nephron, so already have traveled from the glomerulus, right here, into the nephron, and now are going to go back from the filtrate into the blood. And so specifically where the most reabsorption occurs is here at the very beginning of the nephron, what we're going to call the proximal convoluted tubule, or PCT for short. And we're going to have several proteins or transporters that are going to move those things across. What's cool is that we have these symports that allow for a ton of reabsorption of sodium, but it's going to take all glucose, all amino acids, all of the lactate, and then allows for a lot of reabsorption of a lot of reabsorption of bicarb through the buffer system, because it's going to allow for the change of hydrogen and bicarb into CO2 and water, which can be reabsorbed, and then bicarb can get through on the basal surface. Then we just have a solvent drag, which is where water is going to pull calcium and potassium through between the cell. So paracellular, as you can see in this drawing right here. So we have all of those other ones that I talked about using channels are going to go transcellular, and then that solvent drag is going to go paracellular, like so. So that is how a lot of those are reabsorbed. But we can reach a point where if we have too much, for example, glucose in our blood, we can reach a point where there's so much to where it overwhelms these proteins. It's kind of like a revolving door. If there's too much trying to go in, it kind of gets full, and so it will start to leak out into the filtrate, and it'll be found in the urine. That's going to be called your renal threshold, where there's too much and it saturates these proteins here. And something like that happens a lot of times with type 1 diabetics who haven't been diagnosed yet, because they have too much glucose flowing in their blood, and so when it gets filtered, they reabsorb as much of it as they possibly can. But when normal threshold for that is like 200, if they have like say 300, 400, and even more than that, obviously it has to go somewhere, so it's going to be going out into their filtrate, into their urine, which is crazy. Then filtration. If we go back and talk about filtration, there's a lot of different parts to filtration and necessary components to how everything gets filtered out. So where it happens is right here at the beginning of this, and we start with the glomerulus, which is just the buildup of different capillaries and blood vessels here. And that is going to need to have pressure to push solutes and water into the Bowman's capsule, and together that's going to make up the renal corpuscle. We're going to have 4 different pressures: our hydrostatic pressure of the glomerulus, which is pushing in, and then hydrostatic pressure of the capsule, which is pushing out, and then all of the solutes and stuff that are in here, like albumin stuff that's pulling in, and then if there were solutes in here, like albumin, which there shouldn't be, then that would pull in. And all of those together make a net force pulling out at about 10. And if we calculate that, we get a GFR, or a glomerular filtration rate, over a minute at 125 millimeters of mercury. And that is just not selective. It's not picking and choosing what goes through, except for through the layers of filtration. So if I jump over here, I have a little drawing about the layers of filtration. Another little drawing of this nephron. We have 3 layers of filtration. we have first the fenestrated endothelium, which is going to be a barrier for size. So big things like red blood cells won't get through just because of little windows in the blood vessel. And then we have a basement membrane, which is going to be a barrier for charge right there, allowing not negative things to get in. Or small negative things will still be able to get through, like bicarb and chloride, but big negative things like albumin, not big negative things, bigger negative things like, or proteins and stuff like that won't be able to get in because that would mess up the concentration gradient and things like that. And we're going to have these podocytes on the visceral layer of Bowman's capsule, which are going to act as a backup filter. They're going to be for size and charge, but they will basically just be that last filter. And we're also going to have some mesangial cells in here as well, which are smooth muscle to help support that hydrostatic pressure, keeping it at that high level that it needs to be in order to maintain our GFR over here. And that is the process of filtration. Now, we can regulate our blood flow into our glomerulus through a couple different ways, and it can be regulated through like auto-regulation in order to change how high our blood pressure is. So like if our blood pressure is super high, we can make sure that we keep like our hydrostatic pressure even by doing these different things. Basically, if we need to raise our blood pressure, we can initiate the myogenic response. But we also have this other instance called the tubular glomerular feedback, which is just a way that we can increase or decrease our GFR through sensing sodium chloride in the macula densa cell over here, which actually, if this was anatomically correct, nephron, this macula densa cell is like right here. But basically, it's going to sense sodium chloride, which is going to sense how high or low your blood pressure is based upon how much solutes are there. And those, if it's super high, then the macula densa cell would release adenosine, which would then constrict this afferent, and then that would lower the blood pressure. And then same opposite. So if it sends low, then it would want to raise the blood pressure, and so it would do that through releasing nitric oxide, which would dilate the afferent arteriole, leading to an increase in blood pressure or glomerular filtration rate, which is super cool. So then we can talk about clearance, or how substances are cleared from the blood and put into the urine. And that's where I have a little graph and equation down here. Basically, we always are looking for, this is a great diagnostic tool because we can see how, when a substance, so for example, like glucose, if glucose is cleared of the plasma, or how often glucose is cleared of the plasma per minute, tells us things about the person, how healthy they are, what's going on in their body. So normally glucose should always be reabsorbed, should always be put back into our plasma. So if it is a healthy person, it should all be reabsorbed, and so its clearance should be 0 because it shouldn't be cleared of the plasma, if that makes sense. And so what we're looking for here is we have some examples of substances that should be more cleared, or glucose which should be not cleared at all. But then we also have these two substances right here that basically are cleared of the plasma and then found in the urine, but they are still found in the plasma, the same concentration that they're found in the urine. And we're going to talk about those as like a good substance, or a substance that represents our GFR, represents our glomerular filtration rate, because they are not reabsorbed or secreted. They are just simply filtered and then excreted. They just, go right through. And factors for that is that they have to be small and uncharged, freely filtered. They can't be made or destroyed by the kidneys. And they really just tell us the GFR because they tell you how fast your kidneys are filtering, essentially. So we can find that, and essentially this is usually going to be around like 88 to 137 for that clearance level, depending on anatomical variation and male and female variations as well. But then if it's higher than that, it's going to tell you that a substance is being secreted more than it's being reabsorbed. And if it's lower, it's going to tell you that it's being reabsorbed more than it's being secreted. So for example, you can see glucose is being reabsorbed, but you can reach that threshold again to where you start being excreted and not reabsorbed anymore. And that's where you start to be found in the urine instead of the plasma. And same thing on this other end, we have a substance called PAH, which is man-made and given, but it can saturate proteins as well on the secretion side and start to be found in the plasma more than the urine, which is crazy. Then the next thing is just the RAS system again. We've gone through the RAS system a few times, talked about the renin-angiotensin-aldosterone system and how it affects blood pressure. We talked about this in the heart, but it specifically affects the kidney as well. And so we just want to go through it again. I drew a simple version right here, but essentially the kidneys always are just going to detect low oxygen, and that's just through right over here the macula densa cells and the cells that are surrounding it right here. The juxtaglomerular cells are going to sense low oxygen or low glomerular filtration rate, and they want to make sure that blood pressure is high. And so they're going to release a hormone called renin. Renin is going to react with angiotensinogen, and that's going to turn it into angiotensin 1, which is inactive, doesn't do anything. That's going to react with an enzyme called ACE, which is going to turn it into angiotensin 2, which is not active. That can do a few things. It can do widespread vasoconstriction, but also vasoconstrict the efferent arteriole. And when we vasoconstrict the efferent arteriole, if we go back here, that's going to cause the blood pressure here to increase, causing the GFR to increase. It also can go to the adrenal gland, which just sits right above the kidney, and that's what's going to release aldosterone, and that would increase the activity of the sodium-potassium pumps that sit right here, right here on the principal cells, but also in the late distal collecting duct to increase sodium reabsorption, which then would make water follow, increasing the volume of the blood. And then I can also go to the brain, which increases ADH, which would stimulate right here again, increasing water again, which would, increase volume of the blood. And it would also go and tell you that you're thirsty and that you want salt, which would also increase the volume of blood. So all of that would lead to increased blood pressure. Then, basically the last thing with this is that we have a few things that are super cool with our nephron. Basically, we have this thing called the countercurrent multiplier system, where we have our blood going one way and then our filtrate going the opposite way. And this allows for a cool phenomenon because our blood first gets solutes over here, and then it travels around, and then the solutes are what drive the osmolarity, or the osmosis for the water to follow, and the water, or the salts to attract the water. And this basically is a concentration gradient so that we can concentrate our urine down based upon which kind of nephron we are. We have two types of nephrons. We have cortical and juxtamedullary, which can concentrate to different levels right here based on which the ones they are. Humans have mostly cortical nephrons, which allow us to concentrate down to 600 million osmoles, but if we had more juxtamedullary nephrons, we would be able to actually not drink as much water and concentrate our urine a little bit more, which is super cool. But what's cool is that it allows us to concentrate our urine because it establishes kind of like a interstitial gradient to then, if we get to the collecting duct and we release these hormones, then we can, according to ADH and aldosterone stimulation, we can reabsorb that water and aldosterone or sodium to reconcentrate our urine. ADH would specifically do this. We can reconcentrate our urine because of the gradient that was already established in the interstitium in order to reconcentrate our urine, which allows us to reabsorb all our water if we need it and hold on to the water so that we can be healthy, but then our urine would be concentrated, which would make it so that we would have more water, increasing probably our blood pressure a little bit, but also making so we have more nutrients like water and all that. But if we were in a juxtamedullary nephron, we could get down to 1200, and then we could also get down to 1200 here as well. And that's going to be it for a nephron. That's everything. Hope you enjoyed. Have a great night. Bye.